Hello everyone. Today we are going to discuss about the proteins. This is your Manohar. Today we are going to explain about the proteins, how the proteins are going to be formed, how the receptors, what is the difference between the domains, subunit, etc. Okay, so before going to topic, let us ask you some questions. Why hair cannot be digested? What is the protein responsible for the light detection by retina? Why bodybuilders should maintain diet? What is mucoprotein? Role of albumin and globulin in drug transport. What is the amino acid responsible in synthesis of collagen? What is immunoglobulins? What is domains, ligand, subunit, receptors? And what is silk? To address these questions, we are going to cover the biological importance of the proteins, the protein foldings, how the proteins are going to be get folded, how many types of fold, and levels of the protein organization like primary, secondary, tertiary, etc. Next, functional classification. So here we are going to discuss about the functional classification, which is very important one. Next, chemical classification and nutritional based classification. Next, we are going to discuss about the domain, because in pharmacy domains are very important in binding the drug to that particular protein. So here we are going to discuss about the domains. Next, we are going to discuss about different protein families. So proteins are one of the essential building blocks of the human body. They are fundamental constituents of the cell cytoplasm. Daily recommended intake of the protein is around 150 grams. The protein molecules consisting elements like carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, etc. together with the trace elements of iron, copper, iodine, manganese and zinc. Unlike the carbohydrates and lipids, only small amount of the protein is temporarily stored in our body and which can be quickly used up upon demand. So when the bodybuilder builds up his muscle, so if he is not taking proper diet, normally this protein is going to be get utilized. So that is the reason why the bodybuilder should maintain a proper diet to maintain normal physique. Next we'll see what are the functions of the proteins. So proteins almost have all the functions, right from the antibodies, cellular messengers, for movement, muscles, hair, nail, blood, brain, enzymes, cellular, each and every part of our body is made up of protein. Protein can perform various functions like this. So we'll say like how this protein is going to be formed by using the peptides, right? So here by condensation of these two, so here two amino acids are going to be in C terminal and N terminal, so there will be uptaking of these water molecule that results the peptide bond. This peptide bond is responsible for the holding these amino acids together to maintain a long polymer of these amino acids. So there are thousands and thousands of the different combinations of the amino acids that can make up the protein, and that would increase it if each one handle multiple shapes. The proteins usually have only one useful conformational. A protein shape is determined by the sequence of the amino acid. The final shape is called the conformation and has the lowest free energy. Protein foldings. Basically, there are two regular folding patterns have been identified, formed between the bonds of the peptide backbone. So first one is alpha helix. The protein turns like a spiral, a fibrous protein. Example, nail, hair, horn. So I mean to say this alpha helix, this arrangement is provide you the strength that that we can feel in the hair, nail, horn, whatever it is. Next, beta sheets. Normally these are the folds back on itself and it is a ribbon like structure. So normally it is present in the globular proteins. So first discuss about this alpha helix. The formed by the hydrogen bonds between every fourth peptide bond. The alpha helix can either coil to the right or left, can also coil around each other. Coil shape a framework for the structural protein such as nails and skins. So this arrangement is responsible for the strength of the horns, nails or hair, whatever it is. So this is the collagen and elastin. So here we can see the alpha helix which can present in wool, feather, glass, scales in fish, horn and even hoofers. So all this tough material is made up of protein arrangement of the alpha helix. Next, coming to the beta sheets. So there is a ribbon like structure which is form the hydrogen bond between these beta plated sheets. So it can be in two types, anti-parallel and parallel. So anti-parallel run in an opposite direction of its neighbor, whereas parallel run in the same direction with longer looping section between them. So example for the beta sheets is a silk, the silk by which we will prepare the silk sarees, silk cloth, etc. Spider, the web, is made up of beta sheets. This is one of the example for the beta sheets. Next, protein folding. The peptide bond allows the rotation around it and therefore the protein can fold and orient the R group in favorable position. Non-covalent interactions will be held the protein in its functional shapes. These are the weak and will take many holes the shape. So here you can see the ionic bonds and van der Waals bonds and the hydrogen bonds. These specific weak bonds allows the folding of the protein. Coming to globular proteins, so globulin is one of the important protein which is helpful for the transport of the drug from one place to another. So here drug may be hydrophilic or hydrophobic. So depending on, so this protein has the ability to bind with hydrophobic and hydrophilic drugs. So here you can see the unfold polypeptide of the globulin. So after folding of this protein, you can see the outer side is a hydrophilic one and inner side is hydrophobic in one. Okay, when the drug is hydrophobic, obviously it will bind to this particular region. When the drug is hydrophilic in nature, is obviously bind the outer surface of this globulin. So that will helpful for the transport of the drug from one side to the another. Next, levels of the protein organization. So depending on the basic structure, we can categorize the proteins into four. One is primary, secondary, tertiary and quaternary. The primary structure, the sequence of the amino acid in a polypeptide chain. Secondary structure, it is the formation of the alpha helix or beta sheets due to the hydrogen bonding between the peptide backbone. Tertiary structure means the folding of the helicals and the sheets influenced by the R group bonding. So here the conformation of this protein is going to be take place because of the R group. Next, quaternary structures, the association of one or more one polypeptide into a protein complex influenced by the R group bonding. that is called as quaternary structure. So these are the example. So here the primary structure is going to be a simple polypeptide chain, whereas in secondary structure we can see the arrangement of these polypeptides in a helical structure. So here, depending on the R group, so here the function of the arrangement of this structure, folding is going to be take place here. So whereas quaternary, so here more than one tertiary structure is going to be arranged in this, and it will form a quaternary structure. So example for the quaternary is the hemoglobin. Hemoglobin is a globular protein with a prosthetic iron group. Hemoglobin is made up of 4 polypeptides, in which 2 alpha polypeptides and 2 beta polypeptide chains. Each polypeptide surrounds a prosthetic heme group. Hydrophobic interactions between the side groups pointed inwards maintain the structure. The hydrophilic side chains point outside make it soluble. So here the hydrophobic nature is going to be inside and hydrophilic nature is going to be in outside. So here you can see 2 alpha chains and 2 beta chains in which the heme is going to be entrapped. So this heme is normally called as protoporphyrin. So this is the structure of the protoporphyrin, which is going to be present inside of this globulin protein. Next we will come to the classification of the proteins. So basically the proteins can be categorized based on its function, based on its chemical nature and solubility, and based on its nutritional state. Okay, so coming to the functional classification, so here depending on the structure, storage, catalytic, contractile, regulatory, receptor, genetic, defense and transport, we can depending on the functions of this protein we can categorize in this manner. So here structural proteins, normally collagen, elastin, keratin, and transport protein which will transport the material in the body. Normally example is hemoglobin, myoglobin, lipoproteins. Protective proteins, example immunoglobulins, fibrinogens, thrombin, snake venom, bacterial toxins, etc. Then contractile proteins which will helpful for the contraction of the muscles, example actin, myosin and tubulin. Next storage proteins, the ferritin, hemosiderin, gluten, casein and ovalbumin. Next receptors and stimuli, rhodorphin. So rhodorphin is the protein responsible for the detection of the light from corona. The membrane receptor protein and acetylcholine, insulin, etc. Next germicidal proteins, example polymyxin B and gramicidin S. Apart from this, antibodies are also proteins. These are the specialized proteins involved in the defense mechanism. Next, catalytic proteins, enzymes. Normally, all the enzymes are made up of proteins which facilitate the biochemical reactions. And regulatory proteins, so hormones. Normally, the peptide hormones. Hormones mostly may be steroidal hormone which will not belongs to this particular region, but here we are discussing about the peptide hormones such as insulin. Second classification, it is based on the chemical nature and solubility. So further we can categorize into 3 different regions. The simple, so we can categorize as a globular and scleroprotein. Example for the globular is albumin, globulin, glutamase, protamine and histone. So globulin, albumin and globulin is very important proteins for the transport of the drugs. Next, scleroproteins, example collagen, elastin and keratin. So it will has the strong physical strength. Next, conjugated one, so glucoproteins. So here the combination of the carbohydrate with amino acids. Example, the antigens on the RBC, like mucoproteins, glucose in amino acid more than 4%, if it is there, we'll call it as mucoproteins. If the lipid is combined with amino acid, example, LDL, next chromoprotein, a colored substance which bind with the amino acid is called as chromoprotein. If the metal combined with the amino acid, we'll call it as metalloproteins. If phosphorus combined with the amino acid, we'll call it as phosphoproteins. And DNA, if attached with the amino acid, it is called as nuclear protein. Apart from that, we have the primary derived and secondary derived proteins. So this is the glucoprotein, which is responsible for the detection of the blood groups. Carl Landstein sir, who identified this blood grouping in 1901, for which he is awarded Nobel Prize in 1930. Next, based on the nutrition, the proteins can be categorized as complete, incomplete and poor. So based on the source of this protein, so all the essential amino acids can present in the eggs. So hence egg, it is said to be a complete protein source, whereas pulses and cereals will lack one specific essential amino acids. In case of the pulses, so methionine, the amino acid is not present in these pulses, whereas in cereals lysine is not there. So that is why we'll call as pulses and cereals as a incomplete protein source. Next, coming to the poor source, lack of more than one essential amino acid, it is called as poor. So GN deficient of the amino acid tryptophan and lysine. So here these are this is called as complete source of the amino proteins, whereas pulses and cereals are the incomplete one, and this corn or maize is a poor source of the protein. So but in commercially we will see so many advertisement of these, but to be in fact it doesn't have essential amino acids, but it has high content of the fiber. Next, domains. Domains is a basic structural unit of a protein structure, distinct from those that make up of a conformation. Part of the protein that can fold into a stable structure independently. Different domains can impart different functions to the proteins. The proteins can have one to many domains depending on the protein size. So here the alpha helix and beta sheets, so these together form a single polypeptide domain. So this domain is combined with another protein, and this is called as the protein molecule made up of two different domains. So domains are important in pharmacy because so most of the drugs are going to be bind to these domains. Coming to these protein families, it have the similarities in amino acid sequence and 3D structure, have a similar function such as breakdown proteins, but do it differently. Here we can observe the elastase enzyme, which is have the similar structure with the chymotrypsin. So though it is having both similar structure and these two have the same function of the breakdown, but elastase is specifically breakdown or work on elastin, whereas chymotrypsin on the trypsin. Here I mean to say the functions of these different. Multiple peptides. The non-covalent bonds can form interaction between the individual polypeptides that cause the binding sites, where the proteins interact with one another. Because of this, so binding sites are going to be formed. Then subunit. Each polypeptide chain of a large protein is called as subunit. The proteins made up of two subunits is called as dimer. So here this is the dimer with two different polypeptide chains. So here there is a hydrogen bond is forming between different polypeptides. So the conformation of the protein gives its unique function. To work, protein must interact with the other molecule, the ligand, the molecule that a protein can bind, binding site, the part of the protein that interact with the ligand, consisting of cavity formed by the specific arrangement of the amino acids. So this is very important to pharmacy, because most of the drugs work depending on its structure and its binding or either agonistic or antagonistic activity by blocking or by initiating the response of the drugs. Next, antibody families. These antibodies or immunoglobulins are made in response to the foreign molecules, either bacteria, virus, pollen, whatever it is. It binds together tightly and therefore inactivates the antigen. The Y-shaped molecules with two binding sites at the upper end of the Y and the loop of the polypeptides on the end binding site are what imparts the recognition of the antigen. The changes in the sequence of the loops make the antibody recognized different antigens specifically. So this is the structure of this the light chain. So here this is where the antigen is going to bind. It is also called as hypervariable region. So depending on the sequence is going to be bind to antigen. It is going to be done neutralization of this. Next, coming to the questions, why hair cannot be digested if ingested? So because hair it is made up of alpha helical structure, tightly arranged alpha helical structures, though that forms very hard protein that cannot be digested by the proteases enzymes. Next, what is the protein responsible for the light detection by the retina? So here the rhodopsin is the protein which is responsible for the light detection. Why bodybuilders should maintain the diet? Because as like the carbohydrates and lipids, we don't have the storage for the lipids. So when the individual is not taken proper protein diet, obviously the diet is the protein is going to be used up. What is mucoprotein? Mucoprotein is the combination of the carbohydrate and protein. Role of the albumin globulin in the drug transport. So as this protein has hydrophobic as well as hydrophilic nature, so is helpful for the transport of the drugs. What is the amino acid responsible for the synthesis of the collagen? Normally glycine. Glycine is a high content of the amino acid present in the collagen. What is immunoglobulins? These are the defense proteins. What is domains, ligands, subunit and receptors? So these are going to be formed because of the proper folding of this protein and which will interact with some other proteins or hormones. What is silk? So this is the beta sheets, example silkworm and spider. Hope this video is useful. Thank you.